What I want to focus on is the nature of sort of time sequence computation, how the brain organizes computation over time, and how that temporal computation can capture aspects of temporal coding. In particular, Binner mentioned the idea of temporal contiguity, the encoding of temporal sequences in the context of context-dependent spatial recognition. Jocelyn actually mentioned a structure that I'm not going to mention, but which we've been looking at, and that is the retrosplenial cortex, and thinking about the integration of multimodal information. You can think about that as providing information, context-dependent recognition of object-action sequences, high-level semantic evaluation, and all of this will come in the context of hippocampal processing in rodents as they navigate in space. But the concepts, I think, are general, and that is how does the brain take complex time-sequenced information, evaluate that, estimate state, and then use that state to make predictions into the future to guide behavior. And one of the things I'm going to focus on is, again, in a sort of a common theme, the complementary activity and functions that are carried out during active attentive processing, animals navigating in space, keeping track of what's going on, and using that to make ongoing, very rapid, ongoing estimations where they are, where they're going to go. And then resting state activity, when animals are completing tasks, evaluating rewards and goal satisfaction constraints, and are able to, upon reflection, update internal models to better guide future behavior. So active online processing, resting state offline processing, and what we see in particular in the hippocampus and hippocampally related areas, such as the retrosplenial cortex, is that these two different functions are expressed in different, have a different temporal dynamic. There are oscillatory modes that accompany them, and by looking at activity within those dynamics, we gain clues as to how networks not only organize over space or anatomy, and that is the sort of the hierarchical organization or feed-forward organization of computation across circuits, but the organization of computation across time, and then how that interacts with the circuits. So that'll be the overall tack of the talk. And again, I'm going to focus on the hippocampus here, shown the rodent brain, with I apologize for missing Neil's talk, but I'm sure he covered a lot of this. Hippocampus, and spatial navigation, seminal work of John O'Keefe, uncovering the behavioral correlates of individual neurons, in particular pyramidal cells within the hippocampus that carry a spatial code, so-called place cells that respond to individual locations in space. The work in human memory, identifying the hippocampus as being central in the kind of memory or autobiographical episodic memory. We can think of that as memory of context in which we have to evaluate and estimate probable actions. What did we do? Not only what happened, but what happened and what might potentially be our causal involvement. And is the notion of agency, how we try to both evaluate and predict things that happen in the world, and then ultimately try to influence those events by understanding our causal influence in that activity. So bringing all of these things together, place cells, space, episodic memory, and then the idea of predictive causal influence. You can think of this, I think, I'm trying to guess what Neil talked about because I wasn't here. I know he had imagination in the title, but I imagine that it incorporated some of those themes. And that is this kind of predictive evaluation, which incorporated context and some notion of self and agency. What can we do in the world? So to link those two things together, the hippocampus in space, the hippocampus in episodic memory, the idea is that there is a common temporal processing theme, the necessity to both evaluate, to recognize and estimate what am I seeing, where am I now, and then to do some sort of inference to predict or evaluate what would be associated with it, what would come next in the theme of our CBMM challenges, grand sort of challenges. And the approach will be to implant microwire electrodes, arrays of them, into into the extracellular space around individual neurons, record the activity of large populations of these neurons. This just shows raw data indicating single electrodes able to pick out these clouds of individual points. The points are action potential discharges, so spikes occurring. This is accumulated over 20 minutes. So you see patterns of activity around the electrode. You separate those. You can analyze populations of individual neurons. From those populations, here's a representation that's familiar to those in the hippocampal field. John O'Keefe used something like this in his little Nobel presentation. This is an illustration of the property of the spatial firing property of cells, place cells, and that is the property of place fields. Individual points are action potentials from single cells. The different colors indicate different neurons. Here, the yellow cell fires in this location on a top-down view of two environments. We call them mazes. They don't look like mazes in the sense that you're not going to get lost on these things. Here, it's just running from one end to the other of a linear track. This is a C-shaped linear track. Animal goes from one end to the other, back and forth. So the yellow cell fires here, red cell fires at this location, green cell fires at this location. One interesting observation that John made, in fact, in his original discovery, was that these cells not only fire, particularly when animals are running along these kind of circumscribed trajectories or paths, not only do they fire at given locations, but they have the property of directionality, as it was described. And that is this yellow cell will fire when the animal runs through this location in this direction, but not in this direction. That is that they're asymmetric in terms of spatial order. So you can think about that as originally it was imagined, that this reflects the asymmetry of view dependence of spatial location. If I'm at this location, I'm looking over here, I'm obviously seeing you. I'm standing here, I'm seeing what you're seeing, two completely different views, and obviously that would distinguish those two responses. You can think of that as, as faces. These could be like different orientations where you have very clearly, they're related, same location, same underlying identity of that state, but with very different composition of underlying Q components. So different views, different responses. Another way of thinking about that, the temporal contiguity view, is that what distinguishes these two directions is that this location, in this direction, you will go through a series of locations A, B, C. The other direction, C, B, A. It's an order difference. It's how the animals actually go, how they experience space and time that determines these two responses. So over the years, 4 decades of work, I would say that the conclusion is that it really is, it's time, not view. And that is that you can dissociate the specific contributions of differential view and of time order. But it's going to come in an interesting form. You think, okay, so the brain's using one or the other of those two representations. It's not that simple. And that is, when you look at this kind of picture, the static picture, activity over 10 minutes, you lose a very important element of the computation, and that is the dynamics, the time dependence of this. And what we'll see is that these representations change very rapidly over time on the time scale of hundreds of milliseconds and are locked to an intrinsic oscillation, the Theta oscillation. So depending on the phase of an oscillation, you will see very different types of responses reflecting very different types of computations. So just to give you a flavor of the dynamics of it, so that was the static view, averaged over 10 minutes. This is the real-time view. Let me see if I can. You could see this on YouTube if you really wanted. This is just kind of a fun illustration of place cell activity. So these little colored dots, the blue dots here indicate, again, there's one cell that fires here. I was pausing. And I've added a little bit of audio. Animal stops, animal moves. So here's a blue place cell. If you listen very carefully, you would be able to hear this background oscillation, this theta rhythm, which is this kind of very rapid modulation. Animal stops, you see bursts of activity. Animal runs. When the animal's moving, you see the spatial correlate. When the animal stops, you see what appears to be sort of unstructured activity, many cells firing, loss of the spatial correlate. So there are these two states, the active attentive state, animal moving, and then the animal stopping, looking around distractedly, perhaps eating if he was at a goal location. And so during normal behavior, animal very quickly switches between these two. Average transition time between the active and the inactive state is about half a second. So active oscillation, animal stops, half a second later, it's switched into this what I'll refer to as an offline state. So this is just raw activity. If I now take the step of taking that activity and doing a simple estimation, and that is asking where would the animal likely be to see whatever instantaneous pattern of firing I'm seeing. I see the blue cell firing, I guess, the animal's probably at this location. That's where the blue cell has its place field. Of course, we can do this across a population, so we do some very simple Bayesian estimation. And now what you're going to see is these triangles are our guess as to the location the animal would be in order to produce the ongoing activity that we see here. Now, a couple of things that you note. When the animal's moving, the triangles, neural code tracks animal position very well. When the animal stops, code no longer corresponds to where the animal is. You'll see the triangles now associated with those bursts. The triangles would jump across the track. So there are really these two states, active locomotion, where the code corresponds to current location, and then inattentiveness, this offline state, where the code now can represent or express locations that are remote from the animal, are associated, again, with these very different macroscopic states. The theta rhythm during locomotion, and what are referred to as sharp wave ripples, brief bursts lasting about 100 milliseconds of high amplitude population activity, which corresponds to very dense unit activity, lots of cells firing. So here you have a kind of a steady stream of rhythmic activity. Here you have these brief bursts of activity. And one thing to point out is that when you look closely at the hippocampal code, so now I'm going to zoom in. This is now over the course of about 2 seconds. So if we look at activity over 2 seconds, we apply that Bayesian estimation, and that is we ask, every 20 milliseconds, what's the location the animal would likely be to see the pattern of activity across these neurons that we see? And in the dotted line here, you see the animal's actual position. So he's moving here over maybe 20 centimeters over the course of 2 seconds. In gray is our estimate, the Bayesian estimate, where would the animal likely be to produce the instantaneous pattern of activity you see every 20 milliseconds. And what you see is that Bayesian, the estimate, doesn't actually track the dotted line in the animal's current position exactly. You see these little sweeps. It seems to go, and this is the duration of one of these sweeps is about 100 milliseconds. It sweeps from just behind the animal to just in front of the animal and does this 10 times a second. We refer to these as theta sequences. They're sequences of locations. They're brief. They last, again, they span about 100 milliseconds and about 30 centimeters. And if you average them, what you find is the hippocampal representation looks something like this. So this is now, we average across all theta phase lock representations. And this is the relative position, animal's current position. This is behind the animal, this is in front of the animal. This is about 100 milliseconds. So what you see is the representation actually sweeps from just behind the animal to just in front of it. And it does this 10 times a second. So the actual hippocampal code, it's not a positional code, not necessarily even a directional code, that is, what locations that I go through. It's doing very rapidly estimation of where am I now, or where did I just come from, and where am I going to be 20 or 30 centimeters from now. So it's doing this very rapid predictive estimation. If the animal turns around, so if the animal's running here, and then he turns around, this is where the directionality comes in. It's a different set of place cells. So there will be a unique sequence for going this way versus that way. So if in fact you have a track where you have the animal turn around, and as I'll show you, if you have an animal on a track where there's some uncertainty, there's a choice, and that is you put bifurcations into the path. where now the animal has to choose one of two trajectories. This sequence will correctly predict the animal's future path. This tells you where the animal is going to go. Not where it could go, but where it's actually going to go. And so if you think about this theta sequence representation, you could imagine, so again, Benrick mentioned approaches that have been used to probe the function of neural systems using manipulations, in particular the inhibition, try to inactivate a structure. You want to know what something does, you use the classic lesion approach. You can do that. You can do physical, chemical, you can do transient reversible lesions, such as using mucimol, temporarily shut down cells. Well, another approach is to use optogenetics. Optogenetics being a contemporary, refinement of that idea with the added ability to very rapidly and reversibly restrict inactivation to timescales on the order of milliseconds. So you can shut off the system, not for a minute or 10 minutes or an hour, but you can shut off the system for 20 milliseconds. And so this was an approach that a graduate student, Josh Siegel, used in the lab. And the idea here was to ask if we can transiently inactivate, shut off the hippocampus, just during certain phases of this theta oscillation. We might be able to ask the question, what's the difference between activity at one phase and another phase when it comes to spatial navigational behavior? What happens when you shut off the hippocampus, for instance, at this phase versus this phase? So if we could just cut out the activity here, for instance, in this window versus this window, activity that predicts where the animal, or that estimates where the animal is versus where it's going to be, does that actually have behavioral consequences? And so the approach was express an active opsin into inhibitory cells, and that is put in channel rhodopsin, which allows you to optically drive inhibitory interneurons, as shown here, and that is you provide some blue light, you activate inhibitory interneurons over a very brief and controlled period of time, and then the complementary inhibition of pyramidal cells is presumably what's going to carry functional consequences. So we're shutting off pyramidal cells over the time scale of about 25 milliseconds. And he did this in the context of a task, which is actually it's basically a variant of what I described there. And that is a task in which the animal has to make spatial working memory choices. This is a back-to-back T-maze, and the animal starts in one of these two starting arms. It walks up the central stem, and then it has to make a choice. The choice is very simple. It's just go to the same side that you started on. If you started on the left side, go to the left side. The animal then comes back and then is forced into one of these two arms. So it's a continuous spatial working memory task. And we've used this previously because it has interesting properties in that it's behaviorally symmetric. When the animal's coming in this direction, it's still starting in one arm, it's still going into another arm. It's just that in this direction, we choose. In this direction, the animal chooses. So it's the idea of looking at choice and the decision-making process independent of the behavioral consequences of that choice. So dissociating choice from behavior. And so what Josh did, actually, this was something that was really just necessitated by the experimental constraints. You have to apply this. Optogenetic, the blue light. You have to do it at a certain time. So, which phase do you choose? Well, of course, and when do you do it? Where do you do it on the behavioral task? Where do you do it in this oscillatory phase space? Well, you do all the combinations. So, what he did was to pick this 2x2 design in which you would either inhibit activity at the peak or the trough of the theta oscillation. So you have these 280-degree phases. Maybe the peak is actually when it's the peak. That's the operative phase. You inhibit activity then. It's like a hippocampal lesion. The animal can't remember where it is. Maybe the trough is ineffective. So you would have these two contrasts. One phase works, the other phase doesn't work. You could also do it at different behavioral task phases. Previous work that we had done showed that the theta oscillation becomes coherent between the hippocampus and the prefrontal cortex here during this choice phase. Just before the animal chooses the hippocampus and the prefrontal cortex, they phase lock in the theta oscillation. That predicts, if they do phase lock, that predicts correct choice performance. So maybe this is the functional phase. This is when the hippocampus is talking to the prefrontal cortex. This is when inhibition of the hippocampus will matter. Now, it's also the case that for this task, the animal has to keep track of and remember where it came from. And that is, it has to encode information here about the starting location. So perhaps the starting location, maybe the hippocampus actually participates in the initial encoding, that it holds the working memory of where the animal started from. So either of these two parts of the task are important, and the hippocampus could be contributing to either. So the design is inhibit at the peak and at the trough of the theta oscillation, and do it either during what we'll refer to as the retrieval segment, when the animal has to choose, or the encoding segment, when the animal starts. And the thinking was, well, we'll find that, that it's peak phase inhibition during the encoding segment. That's when the hippocampus participates. That is one of those. It's some interaction of phase, theta phase, and behavioral task phase. But instead, what he found was that that inhibition didn't have the anticipated effect, and that is that it did not lead to an impairment of performance. Rather, it led to an enhancement of performance. That when he applied the stimulation, he could actually enhance performance, in this case, by about 15%. So typical performance for these rats, 50% is about chance. You train them up, they'll get to maybe 60, 65%. That's pretty typical. A rat that's really performing well on this task might get up to 80%. And it's interesting that generally these animals, they don't actually reach 100% performance. Perhaps when you're training the monkeys, they will work hard for food to get maximum return. Rats, you might think, oh, they're just not as smart. Actually, they're probably smarter, and that is they're not simply doing the task you ask them to do, but they're constantly evaluating their environment, considering other alternatives. Their favorite alternatives, how do I get out of here? And that is they're constantly trying to figure out, how can I get someplace better? And it's this balance between exploitation and exploration. They're constantly evaluating or checking out options that have not previously been rewarded. And so their asymptotic performance at around 75 to 80% reflects this balance. And that is that, even in a task like this, they're not necessarily driven by goal contingent, these goal contingent rewards. And so getting a 15% improvement is really like getting them to focus. It's like, giving them Adderall or something. Say, look, I know you might want to, explore and check things out, but I really need you to get to that 85%. I need you to really focus on the task at hand. And that was achieved by providing inhibition, shutting off activity at a particular phase. And that is that trough stimulation in the retrieval segment enhanced performance by 15%, got them to focus. And that is, you shut off activity in one phase during the retrieval segment, and they get better. But it wasn't simply that phase, the trough is shutting off activity in the trough enhances performance. We could get a similar enhancement of performance by inhibiting at the peak, but doing so during the encoding segment. So there are really two functions that I had mentioned. Hippocampus has to keep track of where you came from. It also has to communicate that, for instance, the prefrontal cortex, when you get here. And those two functions, encoding and retrieval, were both seemingly being carried out at different theta phases and were utilized at different behavioral task phases. So in this case, shutting off the hippocampus could be good, but only if you did it at the right time and at the right phase. Now, mechanistically, so that's just empirically what we see is that there is this double dissociation between behavioral task phase and theta phase. Mechanistically, What's interesting is if you actually look at the circuit of the hippocampus, so here in CA1, CA1 actually receives two primary inputs. One is a direct feed-forward input from the entorhinal cortex. The other, via this indirect, or what was classically referred to as the trisynaptic loop, goes through dentate and CA3. CA3 provides input to CA1, entorhinal input provides input to CA1. The CA3 input is interesting because it has the property of strong recurrence. And I think, again, I didn't hear Gabriel's talk, but I assume he actually talked about, the idea of recurrent networks and how recurrent architecture can provide a certain computational advantage, such as pattern completion or the ability to do sort of within context estimation. And so the idea that you have a circuit that has this recurrence, and then you have this feed-forward circuit that carries information about sort of multimodal contextual information. And they both converge in CA1. What's interesting about these two inputs is that they're actually separated in phase. These two inputs, they're theta modulated, and they're phase shifted by about 180 degrees. So that if you're in CA1, over the course of the theta oscillation, there are going to be periods where your activity is strongly driven by the entorhinal cortex. This is the cues. It's what I'm seeing. It's what am I hearing? What am I seeing? And then in another phase, I'm strongly driven by inputs from CA3, where CA3, based upon fragmented information, might be doing pattern completion. So I have present state information, and then this kind of pattern completed reconstructed state information. And it turns out that these two phases of inhibition, the impact is when I shut off, I inhibit activity here at the trough. What I'm really doing is suppressing the entorhinal input. I inhibit at the peak, I'm suppressing the CA3 input. So what I'm actually doing, by doing this phase-dependent manipulation, is I'm really controlling which inputs does CA1 listen to. Do you listen to the entorhinal inputs? Do you listen to the CA3 inputs? And inhibiting the entorhinal inputs, it turns out, is particularly effective when it's applied here in the retrieval segment. Suppressing the CA3 inputs is particularly effective when applied in the encoding segments. So you have this picture that hippocampus starts off, it's in the encoding segment, and very rapidly it's switching 10 times a second. There's this theta oscillation. Entorhinal and CA3 inputs competing. Entorhinal input saying, this is where you are. CA3 input saying, yeah, okay, that's where I am, but based upon that fragment of information you have, I'm going to complete that and estimate where I'm going to be. So you're constantly doing estimation and prediction, 10 times a second. Now, prediction is useful here, not so useful in the encoding segment. Where are you going to be 30 centimeters from now? That's pretty much fixed. So there are periods during which prediction, not so useful. So by suppressing prediction during encoding, and conversely, suppressing encoding during prediction, keeping track of where you are here in the retrieval segment, it's not really helpful. There's no additional information that comes. Nothing in this task is contingent upon any difference in location at this position. So just turn off that channel. And that can be done, again, through the separation in phase. But what's interesting is when you now think about these three things, separation in phase, separation of inputs in phase, separation of functions, and then mapping that into this theta sequence. So what we have is the phase, which is effective for encoding, occurs here during the phase which reflects the representation of present location or context. The CA3 input, which is associated with the period which is most effective in enhancing retrieval, occurs here in this predictive component. So again, the hippocampus is doing 10 times a second, taking entorhinal input, estimating present location, and then integrating and incorporating CA3 recurrently driven predictions to estimate where the animal is going to be about 30 centimeters from now. And it's doing that in this phase-dependent manner, which also happens to correlate. If you look downstream, I had mentioned that the result in which on that task, Selective coherence between the hippocampus and prefrontal cortex in the choice during that retrieval segment will predict correct performance on the task. If you look at the preferred phase of the prefrontal cortex, prefrontal cortical neurons also theta phase law, and they themselves have a preferred phase. And that preferred phase, in general, in this case, happens to preferentially align with the predictive component. So in this task, the prefrontal cortex can essentially tune in to different channels, to different phase channels. And when the phase channel that's most useful is one that predicts future location, the prefrontal cortex can use that. Now, one interesting property of prefrontal cortical neurons, unlike hippocampal neurons, is hippocampal neurons tend to have a relatively narrow preferred phase. Prefrontal neurons have a very broad preferred phase. So depending on which neurons you look at, they will actually prefer different phases of the hippocampal theta rhythm. And so I was, in thinking about things like differential preference, cue preference, perhaps, different properties of invariance, view preference, I would say it's likely that those sorts of preferences may also break down in terms of preferred phases, different phases providing certain computational advantages, which is what we see here. Prefrontal cortex likes the predictive information in the hippocampal code. And that's just pointed out here in this result by Matt Jones, in which you see, again, in the same task, preferential coherence in the theta band, which occurs when the animal is going in the choice direction and makes a correct choice. Now I want to just quickly switch to the offline results. I know I'm pretty much out of time, but I'm going to skip anything involving sleep, and I want to switch to this idea of offline processing. A resting state is now reflecting a period during which not only can animals evaluate potential rewards, but they can integrate information about behavior into modifying these models or using reward-dependent learning to alter hippocampal responses and to use that to drive this kind of predictive, goal-dependent, theta-driven activity. So just very quickly, we see in the hippocampus these two different states. During quiet wakefulness, animal stops and rests. The activity switches from this theta rhythmic modulation to this so-called sharp wave ripple-driven activity. And this typically occurs when animals stop, when they eat, when they engage in any activity that does not involve evaluation of consequences to the external world, so-called consummatory behaviors, satisfying internal constraints. And some work by David Foster when he was in my lab, recording from multiple place cells, animal running on a simple maze. What Dave found was, and this is now data of multiple cells over time, a single trial as animal moves from one end of a maze, this is position in time, moves from one end of the maze to another, and then just sits and does nothing for about a minute and a half. And what you see here, these are different cells active at different times because they're active at different positions. This is the spatial or place code. As the animal sits, you see these periods during which many cells seem to be active. This are these bursts of activity, very dense responses of populations of neurons, which if we expand, you see here in the population response of the local field potential, this high frequency oscillation, or what we refer to as a ripple, across multiple cells. We see that many cells are active, but in this case they're active in an order that reflects the time reversed sequence that the animal had run through. So when the animals stop, the hippocampus very rapidly plays through sequences of locations, but they can do it in either a forward or time reversed fashion. And that on long mazes, you find that these ripple events can be grouped together. You get clusters or groups of them, not just single sharp wave ripple events, but in this case, 4 events, which again, if we zoom in on this, 4 of these sharp wave ripple events, which if we estimate, we do this reconstruction, we find that the estimation position will run through an extended sequence, So you can have sequences of sequences. So during these resting periods, activity in the hippocampus is organized into sharp wave ripple events, which can be grouped together to form extended sequences that can span long distances across the maze. And this just shows an example of one of those. I've just slowed it down. So you can see the sequence. It's where the animal is, and it's going to run along this extended path with a 10-meter track. So this is spanning roughly 6 meters. 6 meters of the track. And it's organized into these 4 short segments. Now, when Dave Foster originally made this observation, he tried to link this to the idea of kind of model-free reinforcement learning. And that is the idea of trial and error learning. Like you're wandering through a maze, you get to the end and you find, oh, that was great. You get rewarded after you carry out a sequence of actions that lead to reward. And one problem in this kind of trial and error learning is the problem of temporal credit assignment. How do you give credit to the things that led up to reward after the fact? And this is the idea that an animal walks to the end of the maze, it gets some reward. What you would really like to learn is not that reward occurs here, it's that the locations leading up to that will yield reward. So you would like to spread reward backward in space and time, or to produce a reward prediction error that has this kind of gradient structure. And David's idea was that this quiet wakeful reactivation or reflection upon past events could give you that kind of, could solve that problem, could give you the capacity to map a reward signal, a graded reward signal, onto a reverse sequence. So you play the sequence in reverse, you express a reward signal like dopamine, and then the pairing of that could give you the transformation of this sort of discrete reward and location association with this graded reward profile. So that's reward and reward prediction error. But then there's the other side of it. This is very much like in the Theta story, the idea of encoding versus retrieval. In terms of encoding in the context of reward is like learning about reward, encoding the distribution of reward gradients. Retrieval is like using your reward estimation to do planning. And that is evaluate where should I go in order to get that reward. And you can think about these reactivation events as serving in both capacities. And one clue is that you see these sequences expressed in both time forward and time reverse fashion. You might think, well, time forward, that sounds like planning. It sounds like I'm thinking about where I could go. Time reverse, perhaps that's about learning. That's about mapping reward back in time, learning the reward landscape. And so David Foster, when he started his own laboratory at Johns Hopkins, made another discovery, and that is when animals are seeking hidden reward, in this case in an open field, and you look at these reactivation events, you find that the reactivated sequences tend to point in the direction of expected rewards, and that is that they look something like a planning signal. So we have these 2 signals. We have reward estimation as part of a learning signal, and we have sort of reward evaluation as part of a planning signal. But one thing that's missing is there's no reward signal. We don't know what the reward signal looked like. So Steve Gompertz, when he was in the lab, set out to actually carry out this recording of hippocampal sequence activation and reward signaling by reporting in the ventral tegmental area, which carries large forebrain projections of dopamine, and has been studied in the context of reward prediction error, here shown in the seminal work of Wolfram Schultz, showing that VTA dopamine neurons actually satisfy many of the properties of kind of classical learning theory in terms of reward prediction. And so recording the VTA in the hippocampus simultaneously in this task that I showed you before, what we find is that there are neurons in the VTA which respond to reward. They respond to delivery of reward, they respond to the expectation of reward, that is, respond on approach, respond after delivery, and are modulated by receipt or failure to receive reward. So they pretty much do what you would expect a reward neurons to do. Additionally, during this task, if you look at the firing rate of these dopamine neurons, they show this kind of ramping activity. And that is, once the animal learns that rewards are given at the end, the firing rates after the choice points will begin to ramp up. And that they develop this ramping response in a manner that is consistent with these sort of formal temporal difference learning rules, in particular QTD learning. The dynamics of a QTD learning algorithm predict the dynamics of dopamine response. So it looks like, yep, this is, sort of model-free reinforcement learning in action. But this is during running activity. What about during the stopping, this offline, the reflective period? What we see there is that in the hippocampus, you have these sharp wave ripple events, population activity, individual cells, and now we look at VTA response. So in the gray, you see these are periods during which there's identified sharp wave ripple events. And you see dopamine neurons tend to fire at fairly low rates, but they do seem to be modulated by sharp wave ripples. When you get a sharp wave ripple in the hippocampus, you get an increased firing rate in the VTA. So there's reward associated with reactivation. But it's not just an increase. There's diversity of response. Some neurons will increase, some neurons will decrease, but they are correlated with sharp wave ripple events. So it seems to be consistent with the simple model. But we can do more than simply identify when sharp wave ripple events have occurred. We can ask, what is being replayed during those sharp wave ripple events? So we're going to take those events, we're going to decode the content. And so here's an example of one of these sharp wave ripple events in which there's a decoded sequence spanning part of the maze here. And so we can ask for events that actually reflect sequences in the maze, what is the dopamine response? And we can contrast that with other events. For instance, here is a sharp wave ripple sequence events in which we can decode a sequence. Here's another event which we can't decode. So there are going to be things the animal's thinking about. I want to go to the home cage. I wish I could kill the principal investigator. I don't know what those things look like. Only the rat knows. We don't have any way of decoding. So there are events which have memory content that we can't decode and those that we can decode. So if we compare now dopamine response for events on the maze that we can decode versus replayed events which correspond to something else, we see the dopamine response is not only selective for sharp wave ripples, but it's also selective for sharp wave ripples that replay the maze. It's content specific. The VTA knows what the hippocampus is replaying. But we can go further than that. So for the events which replay the maze, the locations, there are some locations which are associated with reward, others which are not. Does the VTA know when the hippocampus is replaying a goal location, for instance? This looks at the distribution. The red spots here show decoded locations. That is, if we decode all of these trajectories, all the sharp wave ripple events, and we look at which locations get preferential representation in these sequences, we find goal locations in general get preferential representational bias. Hippocampus likes to replay goal locations. But then we can ask, okay, let's look at hippocampal locations that are associated with VTA response. VTA dopamine neurons fire. What are the locations that the hippocampus is actually representing that location? And what we find is that there's a reward bias. So this reflects the extent to which reward or goal locations are overrepresented, the fraction of locations that are associated with reward. So here, for instance, dopamine neurons will tend to be associated with goal locations about 6 times more than any other location. That's this reward bias of about 6. In general, standard replay events, a little bit, almost twice as many of the replayed locations are goal locations. Dopamine neurons really prefer goal locations. But that strong reward bias is task dependent. If we have them running on a linear maze where they're also goals, they're also preferential locations of reward, we don't see any significant difference in the bias. So this enhanced bias is not only specific for goal locations, but it's also specific for tasks in which that goal information needs to be used, and that is tasks in which the animal has to make a choice. So when animals are in a task where they have to make a choice, hippocampus tends to replay locations that include goals, and the dopamine neurons tend to respond when those replayed events include goal locations. And the dopamine neurons that carry out this reward and task-specific reactivation are dopamine neurons that happen to be phase-locked to the theta oscillation during the task itself. So it links back to the original observation that when the hippocampus is running, you have these theta sequences, you phase-lock to the prefrontal cortex, dopamine neurons that also phase-lock to that theta oscillation, in other words, participate in that instantaneous prediction of where the animal is going to go, they will then subsequently, when the animal stops, participate in the reactivation of those sequences and the expected goal contingencies. Even though I didn't talk about any of this, you find that this reactivation, hippocampal memory reactivation, also occurs during sleep and looks very similar, except that when we look at dopamine neuron activity during sleep, that association goes away. So it's not simply that the VTA and dopamine neurons they're an attribute in sequence memory reactivation, but rather they are selectively incorporated in a task-dependent and a task and state-dependent manner, so that reactivation during active quiet wakefulness within a task is different than reactivation during sleep. In sleep, it's reactivation without reward. During quiet wakefulness, it's activation with reward in the context of choice behavior. So when we think about the two functions, theta and sharp wave ripple-driven modulation of hippocampal activity, in which there is this complement of present state and future state predictive coding, which is phase segregated and recapitulated during the quiet wakeful state. And we think about the players that are involved. In this case, the hippocampus replaying sequences, communicating that to the prefrontal cortex, reward information, which may be driven by either the hippocampus or the prefrontal cortex, converging in either the prefrontal cortex and the nucleus accumbens to carry out both reward prediction error-driven learning and reward-driven planning and decision-making. So that's what we see, dopamine activity during reward-driven behavior associated with sharp wave ripple burst, locked to the theta oscillation, dependent upon replay content, and in particular, the precise reconstructed locations on the time scale of approximately 20 milliseconds, suggesting that you have, again, computations which are sort of exquisitely coordinated with very fine time scale, essentially down to the single spike level, and then coordinated across phase as determined by these 100 millisecond or so time windows, which are associated with either the theta oscillation or sharp wave ripple events. And so basically you have sequence memory encoded and replayed, serving roles both in terms of learning, planning, and decision making. And of course the people that did the work.